Hi, I'm Rachel. This video is going to be about neuronal signaling and synaptic transmission. So let's start by talking about synapses. A synapse is a structure in the nervous system that allows a neuron to pass an electrical or chemical signal to another neuron. It's essentially the physical gap between two neurons with opposite ends of the neurons facing each other. So the plasma membrane of the presynaptic neuron on the axon side is very close to the membrane of the postsynaptic neuron on the dendrite side, which allows the neurotransmitters to travel across the synaptic cleft. Vesicles in the axon terminals release the neurotransmitters, and then they travel over to the receptor proteins on the dendrites, or the other side. In the membrane of a cell, you find ion channels, which are proteins that have a pore down their center, and they control the flow of ions into and out of the cell. Another one of their jobs is maintaining and establishing a resting membrane potential, which is determined by the concentration of ions inside of the cell versus in the cell's surroundings. So really, a membrane potential is the difference in electric potential between the inside and the outside of a cell. Usually the inside of the cell will be 70 millivolts less than its surroundings, so the resting membrane potential is negative 70 millivolts. In this stage, the ion channels are closed to preserve the large quantity of Na plus ions outside of the cell and K plus ions inside of the cell. When a nerve impulse stimulates ion channels to open, specifically the voltage-dependent Na plus channels, positive sodium ions can flow into the cell and cause depolarization. This is when the membrane potential starts to increase as the inside of the cell becomes more positively charged. Once the membrane potential reaches about negative 50 millivolts, the neuron can fire an action potential, which is a nerve impulse. The threshold is that level where the difference in electric charge between the inside and outside of the cell is enough to initiate an action potential. The membrane potential keeps increasing though, and once it reaches about positive 40 millivolts, Na plus channels get blocked again so that no more of that ion can enter the cell. Now that the neuron has created an action potential, it wants to lower its membrane potential back to normal. It does this by opening the K plus channels. So since there are naturally more positive potassium ions in the cell than there are outside of the cell, the K plus ions diffuse into the cell, which makes the cell's charge more negative. Eventually, the cell's membrane potential is brought back down to its resting state, and the potassium channels can close. So that's referred to as hyperpolarization. It's really the opposite of depolarization. Just to recap, hyperpolarization is when a cell's membrane potential becomes more negative. This could be due to negatively charged ions going into the cell, or, like in the case of potassium, positively charged ions flowing out of the cell. Either way, hyperpolarization inhibits the release of an action potential because it brings the membrane potential farther away from the threshold. Depolarization makes the membrane potential value more positive and approaches the threshold, so an action potential can be produced. And here's some more vocab on this topic. So, excitatory postsynaptic potential, or EPSP, is a membrane potential in the postsynaptic neuron that makes an action potential more likely. So, an EPSP would occur because of depolarization. And then an inhibitory postsynaptic potential, or IPSP, is just what the name implies. It's a membrane potential that makes firing an action potential less likely. So an IPSP would occur because of hyperpolarization. When the action potential is released, it needs to travel down the axon toward the axon terminals. It's able to do this because the axon's membrane is constantly depolarizing and hyperpolarizing as different segments of the axon each take their turn completing that cycle. So the segment rapidly changes its ion concentrations to increase the membrane potential, then it fires an action potential, and then it returns to the resting membrane potential. This propels the action potential down the axon. That was a lot of potentials. Okay, so now it might be helpful to learn more about the actual structure of a neuron. So we already know that neurons have an axon terminal side, a dendrite side, and in between those two is the axon. Well, also, a myelin sheath is a cylindrical envelope that insulates the core of an axon, and it helps speed up the transmission of these action potentials that we're talking about. So also, a node of Ranvier is a gap in the myelin sheath. When myelin is used to speed up the neurotransmission down the nodes of Ranvier, it is called saltatory conduction. Great, now let's go back to the synapse. Like I briefly mentioned earlier, in each axon terminal there are vesicles, and each of these vesicles encompasses a chemical called a neurotransmitter. The neurotransmitters are released when the vesicle is absorbed into the cell's membrane. The neurotransmitters travel via the synaptic cleft to the postsynaptic membrane. So once they arrive, the neurotransmitters attach themselves to the receptor proteins and transfer their message, initiating the opening of all these ion channels. So this produces a change in the postsynaptic potential, which causes an action potential like we just talked about. Then the action potential that gets produced travels down the axon until it reaches the axon terminal. The impulse causes vesicles to be absorbed into the cell membrane and release their neurotransmitters into the synaptic cleft. The neurotransmitters bind to postsynaptic receptors and deliver their signal, and the process just keeps repeating like that. After neurotransmitters are released from the receptors, they can either be destroyed by enzymes in the synapse or undergo reuptake, which means they get reabsorbed into the presynaptic axon terminal. It's important to note here that only chemical signals crossing the synapses are facilitated by neurotransmitters. If the neurons are going to transmit an electrical signal, then proteins physically connect the presynaptic and postsynaptic membranes, which is called a gap junction, and then the signal passes through this chain. So I'm going to conclude this screencast with one of the many reasons that all of this knowledge is important, and that's because we can apply it to the use of Novocaine. Novocaine is a drug that prevents pain by inhibiting neuronal communication. It hinders the function of nerve cells by binding to their ion channels in the cell membrane. Since the cell can't open its ion channels properly, it can't depolarize to fire an action potential, which means it cannot communicate its sensory information. So the brain never receives the information about the damaged part of the body. If the brain did receive the sensory info, it would produce pain to make the person aware of the damage. But since Novocaine prevents nerve cells from passing on their information to the brain, the person won't feel any pain. That's why Novocaine is used as a local anesthetic for medical procedures like when you're at the dentist.